Okay, so I'll be giving a talk today on doing optical pooled screens for deep mutational scans. So we've heard from a ton of people about a ton of different MAVs, and MAVs from many human protein coding genes might benefit from richer phenotyping. So for one example of, or two examples of proteins that in the last few years have been scanned are P10 and BRCA1, which are both scanned in really great papers in 2018, and they were scanned in cancer cell lines with phenotypes of growth and abundance respectively. And these are two really general phenotypes that can be scaled to a whole number of genes. However, there are genes like lamin A, for example. It's the nuclear laminar protein, and when you have variants in this one gene, you get nuclear aggregates. So that doesn't really fit into the growth or abundance sort of phenotyping. And similarly, even with P10, although there's been activity scans and abundance scans that have been published, there's other phenotypes as well. So for example, when you express P10 variants tagged to GFP in cancer cell lines, especially variants near the N-terminus, you get nuclear exclusion as a phenotype that's potentially related to pathogenicity. And similarly, for both of these genes, when you put them in more relevant, biologically relevant cell types for the diseases they cause, so for example, for lamin A in the context of cardiomyocytes, or for P10 in the context of iPSC-derived neurons, you get different phenotypes. So in the case of cardiomyocytes, you might get this nuclear lobulation, crazy-looking nucleus phenotype. And in the case of P10, you might get these neurons that have a huge number of cytoplasmic projections. And so it kind of underscores the importance of having a DMS strategy that's able to capture a bunch of different phenotypes and potentially deep phenotypes, specifically microscopy phenotypes. And so something that we would really love to be able to do is develop a DMS phenotyping method that might involve saturation mutagenesis and the readout of optical phenotypes like localization or cellular morphology, something that we've heard a lot about today so far, and be able to make single cell measurements of variant effects. So not just do things in batch, but be able to scale to millions of cells at really low cost. And similarly, be able to do this plug and play in different cell types, not just in cancer cell lines, although those are potentially very imageable, but also do it in things like iPSCs and iPSC-derived cells. And so today I'll be talking a little bit about a method that we're working on to try to achieve these goals. So existing in situ sequencing protocols for pooled optical screens are for guide libraries. So there's this really legendary paper by Feldman and Singh et al. From the Paul Blaney lab that's here in Boston, Cell 2019, where they introduced a guide library into cells, one guide per cell, just like we introduced one variant per cell to create this pooled cell library that then they used in situ sequencing to be able to map genotype to phenotype. And so we have a very similar objective, right? So here's a really blown up image of cells that are expressing a library of lamin A variants. You see some cells are aggregated and some cells have a diffuse pattern. So what we would love to be able to do is assign genotypes to these cells and ask which cells for lamin A aggregate and which cells are diffuse. So I'll go over the protocol for the in situ sequencing next. And so in this library, every cell expresses a protein variant that's tagged to a barcode on the same RNA as that protein variant. And so the first thing we do is we fix the cells, and then we convert the RNA into DNA with reverse transcription. And then we hybridize this padlock probe. And the special thing about this RNA is that upstream and downstream of the barcode that's linked to the DNA variant, there's binding sites for this one DNA oligo called a padlock probe that then we can circularize with the addition of a polymerase and a ligase to seal shut. And now you've moved the barcode from the RNA to the cDNA to the circular padlock DNA sequence. So you can amplify that DNA sequence just like you might amplify a sequence in Illumina sequencer to get this concatenameric repeat sequence that then when you unfurl, you can hybridize sequencing primers to, and then perform sequencing by synthesis, just like an Illumina machine might perform sequencing by synthesis, one base at a time. So when you do that, you get images that look like this. They're kind of dazzling. You see 4 different kinds of colors that show up. I've pseudo-colored this, of course, but this is really just taking a Illumina MySeq kit, ripping it apart, and using those reagents to pipette onto cells. So the 4 colors correspond to the 4 nucleobases, and you repeat this over and over again for as many bases as you want. We've done it for up to 12 bases, and it works really well. And so when we first tried this method, remember, this was first developed in the context of guides, and we wanted to use it for overexpression cDNA libraries in the context of DMS experiments. So here on the left, I'm showing you a bar chart of the number of mean reads per cell and the fraction of cells where we had a single dot when we tried this experiment, and we got way less reads per cell and way less cells that had a single dot when we switched from guides to overexpressing a protein variant with tag to a barcode. And you can see that on the left. A lot of the cells in this very blown up image don't seem to have a dot corresponding to a read. And so to solve this problem, I thought, well, we might not be able to increase the capture efficiency of in situ sequencing, but we definitely might be able to increase the amount of the RNA that's present. And so instead of trying to capture the RNA that had the barcode on it directly, my idea was to capture a different RNA, capture a circular RNA. So there's this paper that was written by Sami Jaffrey's group in Nature Biotech in 2019, where they described this really simple method for creating circular RNAs that like increase to up to micromolar levels in the cell. And so you can use a polymerase III promoter just like a U6 promoter to express this small RNA that self-circularizes itself because it's a ribozyme. When you do that, you get these circular RNAs, and these circles, when you look at a gel from the paper, you can't even see the linear RNA band on this gel, but the circular RNA band is hundreds, thousands of times more bright. And so these RNAs really build up to levels of the cell that like proteins would be at. And so when you use these circular RNAs and put the barcode on circular RNA and express the variants on a linear RNA that's separate from that circular RNA, you get way more dots. And so you get a ton of circles, and we kind of have restored now the efficiency of the in situ sequencing protocol and be able to assign reads for every cell and variants for every cell. And so once we do that, we get a huge number of images, terabytes of images, where we have read data and phenotyping data. So we have to stitch and align all of those images together. And then we do dot detection and base calling, just like as done inside an Illumina sequencer. And then we have genotyped something like 4 million cells in a single 6-well plate experiment. So we get this sort of table where you have a bunch of cells. Each cell might have a genotype that we're able to get from the in situ sequencing. And then similarly, we do cell segmentation and then assign each cell a phenotype. And we're currently integrating with methods like Cell Profiler to try to get really high dimensional phenotyping of every cell. Okay, so going back to our application, so recall that we're studying this protein lamin A. Lamin A is really interesting because not just because it has this nuclear aggregation phenotype, but also because it causes a whole bunch of different genetic diseases. So cardiomyopathy and muscular dystrophy, but also progeria and lipid dystrophy. And so what we did is we expressed lamin A variants tagged to GFP in U2OS cells. You notice that the wild type variants don't aggregate in either U2OS or IPS cardiomyocytes. But some of the variants, specifically the ones we've tested that have to do with muscular diseases, seem to undergo this aggregation. And there's this paper from Anderson et al. That's tested many more variants and found that there's an association between myopathy and aggregation. And so now we have a phenotype, and so we have an application for the in situ sequencing method that we've developed. And so now, instead of using Cell Profiler, we decided to do something much more simple for just generating a first draft scan. That simple phenotype is just taking a neural network that we train with human examples of aggregated versus diffuse lamin A localizing cells, and then just scoring every cell as having a lamin A localization that was either aggregated or diffuse. And then now we get this big table, but the phenotypes are just ones or zeros, basically. And then for every variant, we just average over all the phenotypes and report a percentage that's aggregated. And then we convert that to a score via a z-score. So when we do that, we get this map. This is a map of 96 amino acids between positions 178 and 273 of lamin A, and the negative scores correspond to aggregation. So the blue positions here correspond to aggregation on the heat map. And so here's a structure of lamin A from this Onadal paper in 2019. So I'm kind of showing you with the red box here. This is the part of the protein that we're scanning. So this is a homotetrameric structure. So something that you might notice from the scan is it doesn't really look like a ton of other scans you've seen. And that's because lamin A isn't a globular protein. So it's a protein that forms this huge multimeric network on the inside of the nuclear lamina. And so I'll talk you through maybe one or two different things that we saw on the scan. So first of all, something that we noticed is there's this periodicity. So this is just averaging for every position. What is the average of all the missense variant scores of that position? So recall that more negative scores are aggregated. And so as you go across the protein, there's two big things that we saw. One is that there's a periodicity, and the other is that the two different coils, coil 1b and coil 2, have different average scores. So our scan, to some extent, is at least recapitulating the structural features of the protein. And part of coil 1b, there's this heptad repeat. So lamin A isn't just a structural protein, it's also a coiled coil protein. And so there's repetitiveness. And so if you look here, there's a coil-coil dimer, and there's positions A and D on that dimer are residues that are pointing towards each other. And all of the other positions, B, C, D, or B, C, E, F, and G, are all positions that are pointing away towards solvent. And I've kind of highlighted in purple on the scan all the positions A and D, which are the positions that are pointing between the two coils. And you can see that the score is maximum at all of those positions. And that suggests to us that really hydrophobic mutations on the positions that are pointing outward to solvent are the ones causing aggregation. Whereas many of the mutations between the two coils are not causing the aggregation phenotype. And it really also strongly suggests, although we of course don't know this for sure, it's still speculation, that the aggregation phenotype and the aggregates themselves might still involve the protein folded in the coil as a dimer, because it preserves the alpha helical pattern. Okay, so this is one phenotype, but I guess I want to really emphasize that you can kind of extract multidimensional phenotyping. So we did an assay that's very similar to the cell painting assay you heard about before, where we were able to use dyes to highlight a whole bunch of different cellular structures. So we have all of that information in addition to the laminated localization. And we can then strip off all of those dyes or chemically bleach them and then do in situ sequencing for the same cells. So we have phenotyping and genotyping for millions of cells in this one single experiment. Okay, so as a last thing, I'm going to talk about what we're doing next. So I remember that I told you that we sometimes need to go into relevant cell types to be able to get the sorts of phenotypes we want. And so I'm not going to explain this one vector that I put up here, but this is a vector that allows us to express a protein variant library, but also the circular RNA, so it's a dual expression system in stem cells. And so when we cloned 3 libraries of 3 different proteins, so laminin A, but also PTEN, which I talked about the nucleocytoplasmic partitioning phenotype earlier, and RPS19, which has a nucleolar localization versus cytoplasmic localization phenotype. We cloned libraries of all 3 of these into this vector, and then use some of the vector's features for preventing silencing to then put it into iPSCs, and we're able to get iPSCs that were able to express these 3 different saturation libraries of these parts of these 3 different proteins. And so right now we are working on differentiating iPSCs with these libraries into cardios and neurons to ask, are those phenotypes still present in those differentiated cell types, and are there new phenotypes present? And then, of course, doing in situ sequencing in those cell types. And so this is just to prove that we're able to get the in situ sequencing also working in iPSCs. So the power of the circular RNA approach is it really is agnostic to cell type, because these circular RNAs are so common in the cell that you can apply this to basically any cell type you want. And so just to give you a summary, our method is called variant in situ sequencing, or VIS-seq. It enables saturation mutagenesis scanning via cDNA overexpression libraries. It enables scans on things like variant localization and cell morphology, and it really is able to scale not only in cancer cell lines, but also in iPSCs and some iPSC-derived cell types we're testing right now. And then lastly, it scales to millions of cells at something like 10 times lower cost than single-cell RNA sequencing. And so it's really a scalable technology, and we're trying now a couple of different libraries. Okay. With that, I'd like to acknowledge, of course, Doug, who's sitting right up in the front. He's my mentor. And also Nick Bradley, who's a computer science undergraduate student who works with me, who's developed this really big data kind of analysis pipeline to help us analyze all of this data. And then, of course, the UW MSDP and Genome Sciences and CMAP and CAVA grants and consortia that we're on. With that, I'd like to take any questions, please.
Okay, I think we have time for 2 questions. So I see JT, and if somebody else has a question, raise your hand.
Great talk. How are you thinking about cell coverage in your screens as you're moving, especially in variants? We, for our whole genome screens, got something around 100 cells per guide RNA, something on the order of 400, 500 cells per gene. And we find that we're still dramatically underpowered. So how are you thinking about this when you talk about variants as opposed to whole gene perturbations?
Yeah, I think that's a great question. I think that there's no one size. I think that's a great question. No one size fits all answer to that question. So in the laminin A scan that I showed you, we went, because this is the first time we wanted to do a big scan, we went very conservative. So we did on median 880 something cells per variant. In the future, I think I'd like to get away with less, but I would say probably not less than 300 per variant. And I think that something that people don't appreciate, a lot of these optical phenotypes, there's variable penetrance, and they're highly dependent on things like cell density, the cell line, the cell system that you work in. So I think it's really important to make sure you're controlling for as many of those things as possible. but it's impossible to control for all of them. So that's why it's important to try to get, a lot of coverage.
Of course, a huge fan. I'm cheating. I have my own mic. So Lamin A seems like a really cool case to. Oh, I can have two mics.
Oh, you've got already a mic.
Yeah, I have already mic. we can experiment with multiple. I have it off. I have it off. But like you said, from progeria to so many different tissues where it affects, and you get different kind of diseases. Do you guys have a list of variants that are known to affect different tissues in different ways? And one way would be like in one cell type, could you use the different kind of phenotypes that you get in the nucleus to try to predict what kind of tissue-relevant disease? Or you guys have it in iPS cells soon too. Could you do 4 different cell types? And do you need that to get to all the different tissue types and the different variants of different diseases?
That's a great question. I mean, I think that our major motivation for going after multiple cell types was Lamin A, because that it's just one gene that can be scanned so many different ways. There are, and we have, I guess, the list of pathogenic variants that I showed you. But in terms of like gold standard phenotypes in iPS-derived cell types, I think we're not quite there. So there's lots of phenotypes that are documented in cardios. There's the phenotype of aggregation and then other nuclear phenotypes that are documented in fibroblasts. I'm not sure I know a ton about, the progeria field is also vast, so there's tons of phenotypes in a bunch of different cell lines. In terms of gold standard phenotypes, I don't know. But yeah, I think that our motivation is very much to take a region of the protein and that might, or even like many regions of the protein that might have variants that cause all these different diseases, and scan them in like 3 or 4 cell types and see if we can find anything using these multidimensional phenotyping methods based on like cell profiler. But I think that we don't have a good answer yet. And also its mechanism of pathogenesis is really not well understood for any of these diseases. And that also makes it incredibly challenging, I think.
Great, thanks.